Howdy, and welcome to Primary Structure. Now we're going to talk about how we visualize the protein. And here I'm not talking about the assay where we look for a color change on an enzyme-linked secondary antibody, for example, which is really only one type of assay. We're actually asking, how clean is our protein? How many more steps do I need to go through to separate my protein from remaining contaminants? And in order to do that, we use some other techniques, and I want to introduce those in this video. Now, we talked about how we know which fractions, which test tubes in our chromatography steps have protein by looking at absorbance at 280. And so we've seen this slide before. The absorbance at 280 is the absorbance of light at 280 nanometers by the tyrosines and tryptophans in proteins. And that's in all proteins. So that doesn't really tell us whether or not we have insulin or whichever protein we're trying to purify. It just lets us know which of the fractions are worth using our assay for in order to check to see they have our protein. It helps narrow it down, but it doesn't distinguish proteins. In order to separate proteins and really see how clean they are, how contaminated they are, we use something called electrophoresis. Now, electrophoresis is an analytical tool like an assay in that you sacrifice a small amount of your sample to the assay and you don't recover it at the end. So when we take a small amount of liquid from a test tube, that test tube showed absorbance at 280 nanometers, we use that small amount in our assay, like an ELISA, and we don't recover anything at the end. We just know that we can go back to that same sample and carry on with our purification. That's what we're talking about when we talk about electrophoresis as well. A small amount of your test tube that tested positive for your protein using your assay can be denatured, just a fancy word for unfolded, and run on a matrix called a gel that separates proteins based on their charge and size. Now, we can take advantage of that in a couple of ways, but the most common way is to use SDS polyacrylamide gel electrophoresis to separate proteins as bands on a gel. And those bands are detected when we stain them with a dye called Kumasi Brilliant Blue from the Kumasi region of Africa. Now, this dye binds proteins so strongly that you're going to want to wear gloves when using it in the lab. Otherwise, you have to wait for those skin cells to die because your skin is made of proteins, right? All right, so we're going to look at Sds Page, the separation of proteins really based on their size, thanks to a trick, and the trick is in the SDS part of the polyacrylamide gel electrophoresis. And here's how the trick works. SDS, sodium dodecyl sulfate, is a detergent. And that detergent has sulfate on its head group or its hydrophilic part of the molecule. And that sulfate contains two negative charges. Now, why am I going into so much detail? We'll look at the structure in just a second. But the point is that for every amino acid, at least one of these detergent molecules will be affixing two negative charges. And that means that you will turn your polypeptide, which is rich in different charges and different groups, into a polyanion. You'll coat the polypeptide with negative charges, overwhelming any intrinsic charge. And that way, we can separate proteins by electrophoresis in the same ways that we can nucleic acids, which are intrinsically negatively charged, right? So again, it's analytical. We'll take a small sample, about 10 microliters, from a fraction tube, which usually has 10 milliliters. So you're taking 1/1000th typically of your sample. You boil it in SDS and you that unfolds the protein and forms that linear polymer or polypeptide, and you run it on a gel. No protein is recovered after Sds Page. And this is what you get. You stain with Kumasi blue, and then you destain with acetic acid, which is basically vinegar, and you see bands. And these bands these first two lanes are actually known proteins of given molecular weights. And you purchase this commercially, and they separate based on their size, and they stain with the Kumasi blue, and you can reference your favorite protein against the size of the molecular weight standards and see if that's your protein based on size. Now, it looks like lanes 3 and 4 on this gel were before purification, and lanes 5 and 6 are after purification. And there's a striking difference. There's a lot more of our favorite protein, if this is too big for insulin, but this protein, than there is of anything else. Although you can see there's trace amounts of contaminants in the higher molecular weight region. And then it just looks like they've duplicated it across the gel. Now, SDS is a detergent. Here it is. And here it's shown with its sodium salt. So each SDS detergent, and this is found in shampoo and in dishwasher detergent, it's got a charged group. This is the amphiphilic molecule. The charged or polar end head group carries 2 negative charges, which are the counter ion is sodium when you buy it as a dry powder. And then it's linked through an ester linkage to these methylene groups, which are a linear chain that resembles the fatty acid, all hydrophobic groups, right? So this end of the molecule is hydrophobic, this end is hydrophilic, so it's amphipathic or amphiphilic. Those two names are synonymous. Sodium dodecyl is 12, sulfate. Now you can imagine if each one of these binds to an amino acid, you're going to have a polyanion where the 2 negative charges of the sulfate group are going to overwhelm any intrinsic charge of your protein. And for that reason, your protein is going to migrate as a function of its size. Now in this case, in the gel, there's a semi-solid rectangle here that forms a plane of very thin depth, and that has the texture of jello. And the gel is actually like a mesh layer that traps those linear polypeptides. Remember, we unfolded them by melting them at high temperature in this detergent. And so their movement through a field if you have 2 electrodes attached, will be impaired by friction due to that gel structure. So that's what makes the smaller ones move faster. See how the 6.5 kilodalton molecular weight marker is near the bottom, and then the 14, 21, and so on get larger and larger as you go up. And the larger proteins, like this 200 kilodalton molecular weight standard, is near the top of the gel. And it's because of friction that these separate based on size, not charge. Okay. Now this works for proteins that have no covalent linkages, but as you remember from our amino acids lecture, cysteine can actually form disulfide bonds with other cysteines. So this would not be able to separate polypeptides within a protein that are disulfide bonded together. For that, you would need to, before boiling, add a reducing agent, such as DTT or beta-mercaptoethanol. These are two tools, reducing agent tools, to add to your toolbox. All right, so the mobility of the protein in the gel is inversely proportional to the molecular weight. That just is another way of stating smallest goes the farthest and the largest is impeded. And this is what the apparatus looks like. It's the same apparatus that I show for separation of nucleic acids, but typically nucleic acids are separated in a vertical gel. This is a protein gel. Your sample is boiled in SDS, and in this case, they included a stain. We won't go into that just now. You make the gel yourself with a plastic comb that leaves indentations called wells, and that way your samples that are separated from each other. And then you have a glass plate on either side of that gel when you pour it and it polymerizes. The two electric leads that are connected to a power supply, which is not shown here, bring the electrons in this direction, and that gives this lead a negative charge, the cathode, and a positive charge, the anode. Don't confuse this with physics where a cathode produces positive charges. That's not what we're talking about here. We're talking about the direction that molecules will move. Anode means anion loving. Ode means love. An means anion. The anion loving end is the anode, and because anions love it, it must be positive because anions are negative. And vice versa for the cathode. Only the cations love the cathode, and none of our samples are cations, so they all travel this way. Now, if you switch your leads and you hook it up backwards, your sample will go upwards and won't separate. So all of us do that once and then never again. Okay, so when you plot the data, and we don't really do this anymore once it's been proven, we show that there's a linear dependence of the movement of molecules through an SDS Page gel that is linearly but inversely related to the molecular weight. Okay, that makes it a really good tool for separating our molecules and a really nice way of checking at each step in our purification how pure our sample is, whether there are 2 remaining contaminants, 10 remaining contaminants, or we have a really pure sample. Electrophoresis is also used in 2 dimensions using a technique called isoelectric focusing. And isoelectric focusing is actually where the term isoelectric point was first invented. It was invented because it was noticed that when you separate proteins based on their charge, so now we're not talking about SDS Page, we're talking about putting proteins onto a gel that does not have any SDS around and separating them in a gel that was prepared using an ion gradient. The pH gradient that gets set up in the ampholytes that are used, they're like little buffers that move according to their pH, sets up a pH gradient across the gel. And then you add your proteins and you run them in the direction from the cathode to the anode, and they stop moving when their isoelectric point is met. That is, when they reach the pH at which the net charge of their polypeptide is 0, and then they stop. And then you can add molecular weight markers to this end of the gel and soak the gel in SDS and run it again with the anode down here and separate the proteins in the second dimension. And by doing that, you end up separating proteins in a line in one direction and then in another line in the other direction, so that proteins that have the same isoelectric point are likely to have a different molecular weight and be separated from each other. So if you just drew a line anywhere here, all of those proteins have the same isoelectric point but different molecular weights. And this used to be the favored way of really telling if your protein is pure, and also for identifying how many proteins are in a sample. Each one of these could be cut out and then analyzed by mass spectrometry and identified as an individual protein based on its molecular weight. So we won't be using isoelectric focusing. I just wanted to include it to identify the origin of the isoelectric point, that pH at which your protein stops migrating in an isoelectric field, and the idea that you can separate in 2 dimensions and get more separation of proteins. And lastly, we can separate larger assemblies of proteins like the ribosome, which had that 20S and that 60S subunit, and different sizes for between eukaryotes and prokaryotes. You put your sample at the top, and what's at the top of this tube is a low sucrose solution, which has been mixed with a high sucrose solution in a gradient, so that you start low to high sucrose, and the highest density is the highest concentration of sucrose. And then you put that in a swinging rotor and you use centrifugal force, I guess in this direction, and that separates the 2 components, in this case, the red and the blue, into the fast sedimenting component, the heavier, more dense component, and the slow sedimenting or less dense component. You prick the bottom of the tube with a needle and you allow it to drip out, and you have the sucrose, most dense sucrose in the first fraction. The second fraction has the red component, the dense protein component, and then there's a gap, the less dense sucrose region of the gradient that has no protein, and then the blue protein component, etc. And that really only separates very large, very dense macromolecules like the ribosome. Okay, so that leads us to checkpoint 2, protein purification and analysis. Those separation techniques that we covered, which ones can yield protein and which ones are analytical, like the analytical steps of the assay, and the Sds Page don't recover any protein, whereas the fractions, those test tubes that you identified as having protein and assayed as having your protein, those are actually the recovered protein from the purification, and why you would use each step and how it works, that is, which property is being taken advantage of to separate your protein from contaminants using that type of technique. So make sure you can answer these checkpoint questions before you move on to the next part of primary structure, protein sequencing.